thank you for inviting us, Facilis, to participate. What a distinguished group of speakers and researchers, and it's really fun for us to listen to others who share the same passion for 1,4-dioxane and emerging contaminants in general, because it's a problem that the state regulatory agencies are facing, and the research you all are doing helps us do our jobs. So I want to thank you all for that. So first, we'll get a few little overview of the history of 1,4-dioxane in Connecticut from our perspective. First, tell you about how our different agencies are structured. We do work together cooperatively quite a bit, and on 1,4-dioxane is no exception to that. I'll talk a little bit about the regulatory history, and then we have some drinking water case studies to share with you. Thanks, Pat.
Okay.
Yeah, we're good.
It wasn't advancing for me, so I thought I clicked too far.
That's all right. We have slides. That's all that matters. So as Facilis mentioned, I'm with the Department of Energy and Environmental Protection. I work in the Environmental Quality Branch, and we have several duties spread among 3 different bureaus, overseeing discharges to the environment, air, water, discharges to the land, and also regulating waste disposal. We also have groups dedicated to pollution prevention, so certainly 1,4-dioxane falls into that. Myself, I'm based in the remediation division, and we oversee site cleanups. We have remediation standard regulations which set health protective standards guiding the cleanup goals for contaminated sites. And finally, we of course have pollution source oversight. We have the statutory authority to require cleanup of contaminated sites. And we also have authority to require the provision of clean drinking water to sites where potable wells and drinking water supplies have been impacted. So this wraps into 1,4-dioxane. Next, Pat.
Right, and I'm with the Department of Public Health, and our motto is healthy people and healthy Connecticut communities. We are in the Environmental Health and Drinking Water Branch, which contains the environmental health section, which has our toxicologists in it, our private well program relative to today's program. There are also, I believe, 7 other environmental health programs, including radon, lead, and some other things. And I'm in the drinking water section. I'm in source water protection, and I have another slide here. And a little more information on the drinking water section. We are responsible for implementing the Safe Drinking Water Act. We regulate the public water systems in Connecticut. We have authority over proactive laws and high quality water that protects human health. And one of those laws that I wanted to bring up, because it seemed to be a theme over and over when we're talking about 1,4-dioxane, is in Connecticut, we are one of 2 states that prohibit wastewater discharges upstream of surface water supplies. So that has been quite protective for us when it comes to emerging contaminants, many emerging contaminants. I'm going to say PFOA and PFOS are probably not 2 of those because of the different ways that they can get into the environment. But 1,4-dioxane, noting that it comes a lot from wastewater, we are quite protected in our surface water supplies. There are 2,500 public water systems in Connecticut that serve 2.8 of the 3.5 million residents in the state. 500 of those are community systems that serve from the Hartford area, large cities, to a small subdivision just down the street. 2,000 are non-community systems, which could be anything from a coffee shop to a church to a school. We have 150 reservoir systems in our tiny state, and over 4,000 public drinking water groundwater sources. And on top of that, we're looking at approximately 325,000 private wells that serve over 800,000 people in the state. And this is just a picture of our state. It shows the blue is where public drinking water is provided, in pipes in the ground. All those yellow dots are the public drinking water supply wells. The light green are watershed areas. And what I see that I'm missing here are the aquifer protection areas that protect wells that serve over 1,000 customers. In all, about 17% of the state is covered by source water protection areas for drinking water. Shannon?
First, I'll go over a little bit of the 1,4-dioxane history in Connecticut. First, we started hearing a bit about 1,4-dioxane around 2001 when Tom Moore, formerly the California Water Boards, published his white paper on 1,4-dioxane. That, like, okay, is this going to be an issue? A couple years later, EPA Region 1 started poking DEP, telling us that, this is an issue. We have sites, we should start taking a look. DEP works cooperatively, as I mentioned, with the Department of Public Health, where our human health risk assessors reside. And although this predates my involvement, I understand a comparison value was first established for private wells, which DEP had oversight of relative to contaminated sites. And that was first established at 20 micrograms per liter. Subsequent to that, as we started finding 1,4-dioxane at some of our existing chlorinated solvent sites, we asked for that level to be reassessed. And Department of Public Health came back with 2 recommendations: separate action levels for drinking water and also for bathing, showering. 3 parts per billion was deemed appropriate for drinking water for the ingestion route of exposure, and 50 parts per billion was deemed protective for dermal contact for bathing and showering, based on lower incidences of lower risks associated with those activities. Next, Pat.
So in 2012, in conjunction with the unregulated contaminant monitoring rule and request from DEEP, the Department of Public Health created a fact sheet discussing 1,4-dioxane in well water, and Connecticut DEEP began their outreach to the regulated community to let them start to get them on the radar screen so that they understood what they should be looking for. Connecticut DPH Lab created a drinking water method with a method detection limit of 1/2 of microgram per liter. And then in 2013 to 2015, the unregulated contaminated monitoring rule required large public water systems that serve over 10,000 customers, plus a selection of smaller ones to test for 1,4-dioxane in their sources of supply. And in 2020, just back in January or February, the EPA released its regulatory determination 4, which 1,4-dioxane was in that preliminary regulatory determination. EPA determined that they would proceed with regulating PFOA and PFOS. They made a negative determination on several other contaminants, and 2 contaminants, including 1,4-dioxane, they did not make a determination at all, as they are continuing to look at the toxicology.
So where does this leave us? Well, where have we found 1,4-dioxane so far? Pretty much everywhere we've looked. Based on the wide array of uses which past presenters have covered, we're finding 1,4-dioxane at industrial sites. We're finding it in landfills, in groundwater plumes emanating from the landfills and in the leachate. We're finding it at our federal national priority list sites. We're finding it at our state Superfund sites. Also, the RCRA corrective action sites that are undergoing cleanup. And given that many of these previous sites are located in close proximity to many of Connecticut's drinking water resources, as you could see from the map that Pat showed earlier, we're finding it in public and private drinking water wells. So with that, Pat will talk about our case study with a public drinking water well detection.
Right. So under UCMR3, the results for Connecticut, about 330 samples were collected at 41 different public water systems. The total number of detections out of those 330 samples was 23. There was only one detection over 3 micrograms per liter, and that was at 3.6 micrograms per liter. And just so you know, and I believe this was brought up before, when a water utility detects these unregulated contaminants, they do have to put those results and notify their customers through their annual water quality reports, which are also called the consumer confidence reports. So the public water system that was affected by this detection serves approximately 37,000 people, and it has several sources of supply. It has a reservoir system that provides about 91% of its supply, and it also has 2 well fields. One well field has a single well in it, the other has 2 in it. All the sources of supply were sampled under UCMR3, and they narrowed down the detection to 1 well in a well field that contains 2 wells. So we worked with the water utility. What they had done is they changed the operations of the well field in order to reduce the amount of 1,4-dioxane being delivered to the customers. They're doing annual water quality monitoring, and they're required to do increased monitoring if detections are elevated, and ongoing testing at monitoring well locations around the well field. This well field is also impacted by trichloroethylene, and it's similar to 1,4-dioxane. It is only in one of the wells. So we have monitoring data for 2013 through 2019. They should be doing their next round of data collection right now. It includes data for the individual wells and blended samples, if applicable. Water delivered to the customers since that one detection have remained below 3 micrograms per liter. And the 1,4-dioxane levels appear to be diminishing over time. And right now they're at about 0.51 micrograms per liter. And they've had a couple of non-detects as well. Shannon?
Okay. Next, I have 2 short case studies to share with you dealing with 1,4-dioxane in potable wells, small private wells, let me clarify. The first is one of our state Superfund sites I've been involved with for a number of years, located in central Connecticut, Tylerville. This is originally an old site that had been languishing. Chlorinated solvents were discovered in a handful of wells dating back to the early 1980s. And the drinking water action levels for those respective chlorinated solvents had exceedances, and carbon treatment was put in place. Well, as the years went on, additional investigation occurred. We found additional contaminants, including MTBE and chloride from a DOT salt storage facility. And then in the early 2000s, 1,4-dioxane. So in all, we ended up with about 20 private wells with detections or exceedances of the chlorinated solvents, and about a dozen small water systems that also had issues with chlorinated solvents. Next slide, please, Pat. So fast forward, the chlorinated solvent levels were pretty high. And once we started looking for 1,4-dioxane, we found some pretty significant levels. The highest I saw was around 65 parts per billion. The 1,4-dioxane was co-located with the chlorinated solvents. We had multiple suspect sources, although I'm putting my money on a larger former production facility that had lagoons and had degreasing activities going on historically using both TCE and TCA. Although more recent groundwater monitoring, you don't see any TCA. That has a short half-life, it's gone, but we have plenty of degradation products. So as I mentioned, we first had granular activated carbon treatment on many of these wells. Once we found the 1,4-dioxane, the question became, well, will the carbon still be effective? And we were finding that it was not a reliable treatment. 1,4-dioxane was breaking through rapidly, and there wasn't a good rule of thumb. It depended in large part on the influent water quality. Unfortunately, the water quality in general in this area is pretty terrible. A lot of bedrock wells with high iron, manganese, and even some arsenic. So it was presenting a problem. And that was in part why DEEP had asked Department of Public Health, can we see an action level for dermal? Is that any different? Because the carbon was still effective at removing the chlorinated solvents, and it was actually cheaper for us to maintain the carbon to treat the chlorinated solvents and then provide bottled water to these individual residences to give them potable drinking water. In general, the 1,4-dioxane levels were always below 50 parts per billion when this program started. So that's what we did for many years. Other issues I saw with the carbon treatment, we'd have, say, 10 parts per billion in the raw water, and in maybe 12 parts per billion in between the two filters, because typically you have 2 carbon filtration canisters in sequence. And then in the finished water, an even higher concentration of 1,4-dioxane. So we were getting sloughing of the 1,4-dioxane, whatever was building up on the carbon, being displaced by the chlorinated solvents that had a stronger affinity for the carbon. So after a nearly 30 some odd year history with this site, 1,4-dioxane was sort of the straw that broke the camel's back, and carbon cannot be the long-term solution for this site. We have solvents and likely NAPL and fractured bedrock here. A permanent drinking water solution was needed. And we have a water main project that is pretty much near completion, just tying up some loose ends at the moment at a cost to the state of $7.3 million. Next slide. The other site I was going to talk about, Ashley spoke a little, not Ashley, Michaela gave a good summary. The Durham Meadows Federal Superfund site. I'm not the project manager for this one, but Tylerville and Durham are quite similar. Durham had the pleasure of having 2 responsible parties identified. But again, long history of chlorinated solvent use, lagoons, discharges to groundwater, and initially carbon filtration provided, later 1,4-dioxane in the early 2000s detected. The former project manager on this site had done some testing of different types of carbon and determined that coconut carbon worked somewhat better. So we had been using that, but eventually the same procedure came to pass, where we provided the belt and suspenders. You have your carbon for treatment of the chlorinated solvents, and we're providing you bottled water to protect for the 1,4-dioxane. Next slide. These are some of the statistics, and they have a longer water main currently being installed as part of the remedy for the site. 5.5-mile water main extension plus nearly 1 million gallon storage tank being constructed. Most of the water main is now in, but the individual service connections are needed. And these projects aren't cheap. This is $24 million. The state shares 10%. And we're looking at substantial project completion by the end of next year. Next slide. So this sort of wraps up our quick summary. These are some of the challenges state agencies are facing. Health risk communication, as we continue to find these newer contaminants that weren't on the radar previously. how do you convey to people that their drinking water that we told them was being treated and was clean is now no longer clean and you need something else? 1,4-dioxane is especially difficult to treat. I'm, dare I say, I'm somewhat more concerned about it than with PFAS. At least with PFAS, you can treat it more easily. 1,4-dioxane, you need the advanced oxidation processes. And this is not really available right now on a residential scale. What do we do about residences located in areas where public water isn't available, where there's not a good spot to put in a community well because you have a wider groundwater pollution issue? These are some of the things that concern me with 1,4-dioxane and drinking water. Pat, any concluding thoughts as well?
I think you wrapped it up quite well, Shannon. And if there's any questions, this is our contact information, and we can share these slides with Vasilis so he can share them with everybody else too.
All right, well, thanks, Vasilis. And yeah, sorry everyone for crashing the party here. Let me see if I can. I promised to make this brief. I just want to highlight a few major points here, and a lot of what I was going to present has already been covered. So for the sake of time here, I'll just sort of jump to the chase. And primarily what I want to do is just throw a bunch of data at you for perspective and I guess some context in terms of, the way the DoD is managing 1,4-dioxane and how these things that we discuss at these type of venues really have the potential to really impact programmatically, the way we conduct our restoration business and the types of dollars that are at stake here. So by way of introduction, I'll just say here that, what's already been covered, 1,4-dioxane, of course, is this rather volatile constituent in pure solution, but, due to the copolarity, does not volatilize from water very efficiently. So consequently, people like Detlef have to heat his chromatography columns to get things off in a GC mass spec. Similarly, vapor intrusion is just not something we consider with 1,4-dioxane. And I'll just point out that the major references that are out there that kind of summarize soup to nuts all of the technical elements of this 1,4-dioxane problem are coming out with new editions, especially the 1,4-dioxane book. And I guess I'm kind of biased to give it a plug because my wife Janet wrote the toxicology chapter. Having said that, moving on to some data. This is everything from our program thus far. This is every groundwater sample APCEC has ever taken. We're over 60,000 samples strong at this point. And, truncating everything to the year 2000, it shouldn't come as a surprise to you that we've gotten better. Our commercial laboratories have gotten better at quantifying 1,4-dioxane over the years. And the limit of quantitation presented here is just derived statistically by me based on all these data in terms of how low we can quantitatively measure 1,4-dioxane in groundwater without having a estimate JFlag associated with it. And it shouldn't come as a surprise that as we get better and better at measuring 1,4-dioxane at these ultra low concentrations, well, I say ultra low, it's relatively low. It's not in the PFAS realm of low, but it's certainly low by our conventional contaminant standards. But the better we get at measuring 1,4-dioxane at these dilute concentrations, surprise, surprise, the more efficiently we are able to find them, and our frequency of detection, FOD here, has increased significantly and steadily since 2000 and even before. And I attribute a lot of this to discovery of really low-level large dilute 1,4-dioxane plumes that are associated with all of the other legacy contamination that's out there that we've just been more attuned at investigating in recent years. But again, to put all this in perspective, looking at this in the context of where we are with other contaminants that the Air Force spends a lot of money remediating, a lot of this has to do with our analytical capabilities in terms of how low we can go. But by way of mass, there's much more of this 1,4-dioxane problem out there than there is PFAS, and it's always sort of surprised me, why this PFAS problem has exploded the way it has, and the 1,4-dioxane problem has really lingered on the shelf for a lot of years and really hasn't sprouted legs until now, or at least not as exponentially fast as the PFAS problem has. But relatively speaking, there's more 1,4-dioxane out there at these industrial haz waste sites than there are, semi-volatiles and PFAS. Contemporary sources, I'll just give a plug for the ITRC team that's investigating or comprising the tech reg for 1,4-dioxane. There's a really nice history and use chapter that's in production that goes through all the contemporary sources, and suffice it to say that, you know, the vast majority of the 1,4-dioxane we see in surface waters, and things from, you know, wastewater treatment plants, from, you know, discharges of, you know, household sort of sources, is mostly the result of this ethoxylation byproduct source. It's 1,4-dioxane is produced sort of inadvertently in a number of different manufacturing schemes, and there's ways to do that efficiently and to avoid the production of 1,4-dioxane in those processes, but it's extremely expensive. And, you know, industry's been really slow to adopt those new engineering practices to avoid this process, although the new TSCA rules are sort of starting to force the issue. You've seen this map. You know, I'll just point out, I don't want to steal Dave's thunder, he's speaking later, but, you know, there's some interesting differences between surface water and groundwater sources for where these PWSs are obtaining their water from. And I think that has a lot to do with the differences between the contemporary sources and the legacy sources. With regard to the legacy sources, you know, I just want to point out, everybody sort of points to the fact that all of this 1,4-dioxane from, you know, historic solvent applications are from TCA, and that's probably true, but I think it's underappreciated that, you know, every single chlorinated solvent ever manufactured and used for the purpose of metal degreasing, you know, reacts with zero valent metals through the the dehydrohalogenation pathway or the beta elimination pathway to produce these acids and these corrosive byproducts. And so the chemists figured this out a long time ago, World War II era, and they started manufacturing solvents, every single solvent with inhibitors to prevent those processes. And yes, the composition of the solvent does, and the chemical properties of the solvent does clearly affect, you know, what inhibitors are relevant and how much, you know, inhibitor is necessary to prevent those reactions. But it's absolutely unequivocal that throughout history, every single chlorinated solvent manufactured for the purpose of metal degreasing has been inhibited. It's just a question of with what. And if you go back and look, you know, people overlook the fact that, you know, TCE, even modern TCE, before it was, you know, outlawed in this country at least, you know, was inhibited. And there's all kinds of proprietary, you know, ways to do that. And that's really what sets these different manufacturers apart in terms of, you know, their product is not the TCE. The TCE is the same molecule in all these formulations. It's the other stuff that they put in these formulations to prevent these corrosive byproduct formation and their suitability for different applications. And unlike TCA, that was, you know, where the inhibited 1,4-dioxane inhibited TCA, where Dow had the monopoly throughout the production history of TCA, there are many different companies that have manufactured and sold TCE formulations that have been inhibited with something. And again, what sets these companies apart, their products apart, are the other things they put in it. And just from a DoD perspective, you know, from the early 40s, you know, when the World War II, you know, mass aircraft production era started, the military produced a military specification for TCE for the production and maintenance of those aircraft parts. And it was required by specification that every last molecule of TCE be inhibited. It does have language about the amine type and et cetera, but it was not specific and allowed some flexibility for the manufacturers, because, I mean, at that time, you know, the US military couldn't get their hands on enough of this stuff. So they would take it from anybody who could manufacture it, but it had to meet these military specification requirements, which were really performance-based. They didn't have a lot to do with the chemistry per se. It just had to react or not react with metal parts in a measurable way that was defined by these regulations. So bottom line is, every last drop of TCE used in this country, if from the military's use of TCE, if not the country at large, for the purposes of metal degreasing, has been inhibited. It's just, again, a question of with what. And I like to point this out because a lot of people, you know, talk about the TCA degradation products, but really, you know, 1,1-DC, yes, is the hydrolysis product of TCA, but it's also the reductive dechlorination product of TCE, or a reductive dechlorination product. And if, you know, you go back to the 90s when everybody was doing pure culture experiments with TCE and pure solution, looking at, trying to define these pathways. A lot of 1,1-DC, materialized in those microcosm experiments, and all that data is published. And I just take exception to the fact that people refer to 1,1-DC as a surrogate for TCA, because that's not a fair assumption. And when we've looked at our portfolio of data, and this is kind of outdated, the 2000 numbers from some years ago, we're over 3,000 strong now in terms of our 1,4-dioxane. The spatially discrete wells, we got a lot of repeated measures, and that was the data I showed you earlier. But in terms of spatially discrete monitoring wells, we can kind of slice and dice all the 1,4-dioxane in terms of what it co-occurs with. And no matter how you look at this, whether you approach it from an epidemiological perspective, which was the case that we, back in 2012 when we published all this data, if you approach it more like an engineer and try to account for all the degradation products, you just simply can't attribute all of the 1,4-dioxane that's out there to TCA. It definitely co-occurs with TCA, that's unequivocal, but there's a good significant fraction of the dioxane that's out there that just has nothing to do with TCA and or 1,1-DCE in a lot of instances. And so therefore the assumption is that there's some other source of 1,4-dioxane, and there could be situations where things like de-icing fluid and other non-solvent sources have been, the result of some of these groundwater plumes. But I would say in a fairly large number of instances that are underappreciated, I think throughout industry, TCE is probably the culprit. And again, this is complicated by some transport, chemical specific transport and potentially differential degradation processes. And I think Dave will probably cover some of that, if not Shaylee. But the point is, no matter how you look at it, in my professional opinion, there's a lot of 1,4-dioxane out there, at least at these legacy industrial sites that, in my opinion, originated from TCE. And you don't have to take my word for it. There's case studies out there that are published. People kind of dance around this issue, I guess, because they don't want to create controversy. I guess I'm not, a fan of that. It is what it is, and people should speak their mind. And point in case, this is a really smoking gun case study, and it unequivocally demonstrates TCE as a source of 1,4-dioxane at a waste site in Germany. But there's others. Just to mention health effects real quick, and this has already been talked about, and this is, of course, in my swim lane, but I'll just say that cancer drives all of our risk assessments, and of course, all of the data that has been used to define these reference doses and these cancer slope factors are generated from animal studies, and of course, that has its own inherent sort of limitations. Everybody's referred to all these different state values that have all this variability out there. I won't belabor the point. I think this figure or something similar has been illustrated. But what I want to point out is that, a lot of this variability is just attributed to different science policy decisions. And, we can argue about the relevant risk range to make decisions, and that's fine. But in forums like this, when academics get, around one another and start talking about all this mutagenicity stuff and, whether there's a threshold effect or there's not, it's usually talked about in the context of science and academic, kind of exercises. But the real world reality of this is, if you adopt the Canadian model where there is a threshold effect and you use their values to repopulate all these maps that have been produced throughout this, this forum, all the red dots go away. And that's because, what's out there in the environment in reality are these, fairly low levels, as I've already demonstrated. And I don't have the reference line on here to show you, but if we move away from this linear low dose extrapolation and the current risk paradigm in the US and adopt the Canadian model, there's essentially no more to talk about. And I think that's relevant to keep in perspective here, because the amount of remediation and the amount of risk management that is taking place with regard to 1,4-dioxane, and it's not nearly as big of an issue as what's emerging for PFAS, because we have this natural attenuation the pathway that is emerged as relevant. And I think Dave, again, will talk about that. But where we are taking active remediation efforts to address 1,4-dioxane, it's to mitigate concentrations that the Canadians would not perceive as adverse or to represent an adverse condition. So I think that's important to keep in mind, because we're spending an awful lot of taxpayer dollars to mitigate a problem that may not exist depending on what side of the spectrum you fall on with regard to the toxicology. With regard to AFCAC, I'll just say this sort of in conclusion, this is our portfolio. This is a three-way proportional Venn diagram. So it represents, the overlap between our TC and TCA sites and the 1,1-DCE sites. And where all of our 1,4-dioxane occurs within this, which again is like 95+% of it, we are taking efforts to get our hands around all this 1,4-dioxane. And that started years ago with a series of efforts to conduct confirmation sampling. So 2 or 3, monitoring wells, we would, sample for 1,4-dioxane, spend the money for the heated purge and trap isotope dilution method to get good sensitive numbers down at the low dose range or low concentration range. And then where we had detections that are, relevant to whatever state scenario, we conducted then step outs to do full scale delineation. And we're still in the process of doing that. We've been wholly, sidetracked by the whole PFAS thing, but, we are marching our way through the program with that. And a number of sites we are undertaking, remedy reevaluations, especially for sites where we've been addressing these chlorinated solvents with things like air strippers that don't have the potential to remove 1,4-dioxane, or we're going back and retrofitting all of those systems to implement tertiary treatment to either oxidant-based, biological-based, absorbent-based. We have a number of different scenarios and different examples where we've installed, additional treatment trains to mitigate dioxane. But anyway, we're on top of it and we're slowly working our way through all of this. I'll just say that in conclusion, there's a number of sources out there. It's clearly a significant issue. It probably merits more attention than the PFAS problem, but that's a subjective opinion. We are, really battling with all these transient state regulations. And I'll just mention that, when states say that they have values on the books, that's not necessarily, legally binding in all cases. A lot of them are guidance, some of them aren't. Some states that say they are legally binding, our attorneys, beg to differ. And it has to do with the process by which these states take their promulgation of these regulations. And it seems to be way more complicated than it should be, but nevertheless, it's something that keeps our attorneys gainfully employed. I do fundamentally believe that the scale of the problem at our chlorinated sites is bigger than what can be explained by TCA. I certainly, prescribe to the notion that the majority of 1,4-dioxane at these historic haz waste sites is probably the product of TCA use, but that alone just cannot explain the totality of it comprehensively. And then I would say we're painfully slowly, executing our program to get our hands around all of it. And, there doesn't seem to be a whole lot of off-base issues with 1,4-dioxane like there has been with PFAS. And I think that has to do with the fact that there is this natural attenuation mechanism that's operative, especially as you get down gradient in the more aerobic portions of the aquifer at these contaminated sites. But I'm happy to report that we're working our way through it. And, we're trying to take an earnest effort to get our hands around the whole thing and take responsible action where it's necessary. So with that, I'll turn it back to you, Vasilis.
Thank you, Vasilis and the team, for inviting me to share some of our research. It's an honor to be participating in this group and among the stellar cast of speakers that you assembled. So I will just talk about some of the research that we've done. My lab at UCLA works, we are civil engineers, microbiologists. Some of our research focuses on biotransformation of emerging contaminants, including 1,4-dioxane, PFAS, other stuff. We also do a bunch of omic stuff in these food, energy, water systems, and then soil, plant, gut implications, various types of omics tools, and then also applications and implications of nanomaterials in their interactions with microbes. So I'll just be touching on some of the highlights that Vasilis asked me to cover, but we've published over 25 papers. I'm happy to share any of those publications with you, but I just listed them here so you would have them. I don't think I need to repeat this about 1,4-dioxane and its health effects and in environmental fate and transport. That's governed by the chemical properties of the substance. I'll skip that. I'll just say it out up front. Yes, for many years, dioxane was not considered biodegradable, significantly biodegradable. When I started my PhD in 2001 at UC Berkeley, I think there were just 1 or 2 studies by Rebecca Perales, who was then at a company, and Mike Hyman, who's going to speak after me, had some evidence with 1,4-dioxane and other ethers being biodegradable. But now the evidence is growing. There are several reports of pure cultures of aerobic bacteria and fungi that have been shown to degrade dioxane. Unfortunately, there is no real evidence of dioxane degradation under anaerobic conditions. And it is possible that that could happen, but we just don't know yet. There's co-metabolism, in which bacteria that use some other substrates as energy sources are able to also biodegrade 1,4-dioxane. There's mixed communities that are found in industrial sludge. I do a lot of work. Others have reported treatment trains, which include one or more abiotic processes and one or more biological degradation systems. There's a report out of Georgia Tech on Fenton reaction. We've been working with, there are other field efforts, pilot scale, full scale field efforts in situ and ex situ that show dioxane removal, dioxane degradation. And there is limited but ever-growing evidence that dioxane can be naturally attenuated in groundwater. And I'll show some data that we work with. So speaking of enzymes in many of these systems, what the common thread is that similar to the studies evaluating carcinogenicity of this compound where liver P450s were implicated, similar systems found in prokaryotes, bacterial monooxygenases, have been shown to catalyze the critical first step, the rate limiting step of breaking the cyclic ether structure and then setting off a cascade of downstream biochemical reactions. So monooxygenase containing bacteria, aerobic bacteria, whether it's metabolism or co-metabolism, have been confirmed. Many publications by me and colleagues all over that it seems very convergent that these are monooxygenase enzymes, multiple lines of evidence, really solid evidence of that. So that's been published before. And like I talked about, maybe this group doesn't need that kind of background, but just if some of you are not microbiologists, biodegradation processes could be supporting growth of the microorganisms that carry out these reactions. They generate these enzymes meant for the contaminant itself, serving as the growth substrate, and we call it metabolism or growth supporting. So there's some examples of bacteria, for example, Pseudonocardia dioxanivorans that I isolated and characterized. And then in other cases, there are bacteria that would produce enzymes, in this case, say monooxygenases, for biodegrading a different substrate that yields energy or carbon for the growth of the microbe itself. But just by sheer accident, the same enzyme can also degrade a different compound, in this case, dioxane, or many other xenobiotics. And we call that co-metabolism or a non-growth supporting metabolism. But both of those processes have been well characterized in aerobic bacteria for dioxane biodegradation. I'm mostly going to focus on metabolic growth supporting processes because I know the talk right after mine, Mike Hyman. He is the ultimate expert in co-metabolism of various organic compounds, halogenated ether compounds, petroleum hydrocarbons, all sorts of emerging contaminants. So I'll just let him focus on that. With respect to biodegradation pathway, we talked about how monooxygenases are involved in the initial ring cleavage and hydroxylation. This is pathway that I've published in 2007, then some updated a few times since then, but the overall skeleton remains the same. So if you focus on the top dioxane molecule right here, it gets hydroxylated, different intermediates. This compound right here, beta-hydroxyethoxyacetic acid, that was mentioned earlier by Mary and others as the terminal metabolite in toxicology studies, at least in human and mammalian studies, where that was the primary metabolite of dioxane exposure. In many bacteria, especially those that can use dioxane for growth, once we pass this point, the carbon dioxide, so nearly stoichiometric amounts of dioxane is mineralized to carbon dioxide in these studies. So when we were talking about dioxin, and again, following up with Detlef's and Hunter's talks here, that dioxin was used as a stabilizer for chlorinated solvents, different chlorinated solvents. As a result, it has found co-contaminant of chlorinated solvents. And conveniently, monooxygenase expressing bacteria, some of those that have been shown to biodegrade dioxin, have also been shown to metabolize or co-metabolize several chlorinated solvents. So that made our study of understanding these biodegradation processes in mixtures really helpful. We did some work with Dave Adamson, who's also going to talk later today about co-occurrence and implications of degradation and natural attenuation and other aspects of dioxin. So I won't cover that. I'll jump right into some of the lab data that we produced. So what you're seeing are 4 plots with time, 24 hours on the x-axis, and dioxin concentrations in micrograms per liter on the y-axis. And here we are using Pseudonocardia CB1190 strain to biodegrade dioxin in the presence of various concentrations of chlorinated solvents. So 111 TCA, TCE, cis-TCE, and 11 DCE. Bottom line is there is a dose-dependent inhibition, and 11 DCE is one of the strongest inhibitors of dioxin degradation that we've reported so far. Not only in pure culture, we actually did some of these things in several different microcosms and more controlled field scale studies in situ as well as ex situ reactors in which we took, I'll talk about the microcosm first so that it gives a little flavor of what we are looking at. So we set up microcosms, different treatments, undisturbed, sort of as is, what we call a natural attenuation simulation. We also bio-augmented with Pseudonocardia dioxinivorans, and then tried to do with and without chlorinated compounds, top-down, bottom-up type of an approach. In this microcosm, these were samples collected from a contaminated site. So concentration of dioxin was about 500, 400 micrograms per liter. Unlike the previous study, and that was published in Environmental Science Technology, I've paid for open access, so that paper should be available to anyone, even without subscription. This is also from the same paper, but this is 10 weeks rather than 24 hours. So what we are seeing here are number of controls, the tan and the brown and the gray lines, sterile control, other in which we didn't add anything. There was really no removal of dioxin at that particular site. And I'll get into why we think chlorinated solvents were inhibitors for that process. But when we bio-augmented with CB1190, and that's the blue open squares here, in about 4 weeks, we saw significant removal of dioxin and much lower, and it looks like high here, but that's because we had an analytical issue. We were not able to use our GC mass spec, but only the GC with a flame ionization detector. So I'm pretty sure we went quite low on that experiment, but these were the numbers we measured. When we removed CVOCs, those are the green triangles, we immediately saw rapid degradation of dioxin in about less than a week. And that sort of back calculates to the fact that chlorinated solvents are inhibiting dioxin degradation. We also constructed mixtures with, building up representing different sites that we had obtained contaminated soil and water from. This is again a shorter term study, unlike the 10-week one, but we wanted to eliminate one compound at a time and then also use mixtures of chlorinated solvents. And bottom line is, it doesn't really matter which other chlorinated solvents are present as long as significant concentrations of 1,1-DCE are present, in the thousands of microgram per liter, hundreds of micrograms per liter. It's almost a deal breaker for dioxin degradation. So the details I'm happy to discuss offline. But I'll stop there with the chlorinated solvents. I wanted to bring awareness of other types of co-contaminants of dioxin, since dioxane and chlorinated solvents are used for degreasing vapor degreasing of metallic components. There are in the environment metals, copper, zinc, chromium, iron, manganese, hexavalent chromium was a big one, nickel that are found as co-contaminants of dioxane in surface waters, in groundwaters, drinking water supplies, industrial wastewaters elsewhere. And it's important because we're talking enzymatic degradation of this organic compound, and many transition metals serve as cofactors of these enzymes. Here's an example of methane monooxygenase. Similarly, now that we've characterized dioxane monooxygenase and others, they definitely have an iron or a copper active cluster at the cofactor at the active site. So then we looked at the degradation of dioxane in the presence of various concentrations. And I recognize these are high concentrations. We do have other data, but this produces the most dramatic contrast that I just hope would be interesting in this kind of a setting. We would normally think copper is an essential nutrient for prokaryotes, as many other organisms. But in this case, the presence of copper actually was one of the strongest inhibitors of dioxane degradation activities. Cadmium was also there, zinc, nickel, hexavalent chromium I'm not including in this plot, but we recently published a paper in Stoughton last year. That was also very surprisingly, so we think chrome is much more toxic than copper. The results were reversed. Hexavalent chrome was actually not as inhibitory as copper and cadmium in this case. But luckily, there is a lot of natural organic matter, various ligands that are available that can complex cations, transition metal, and oxyanions that can mitigate toxicity by these heavy metals, these trace transition metals, towards the enzymes that catalyze dioxane degradation. So I'm going to break this presentation in two parts. The fundamentals and the co-contaminants are summarized here, and then for the rest of my time, I'll talk about biomarkers and natural attenuation. So basically, chlorinated solvents did inhibit dioxane biodegradation, with 1,1-DCE being the strongest one. And with respect to mechanisms, I think the universal stress protein and ATP yields were some of the reasons that help explain the inhibition. Trace metals, yes, some of them were inhibitory in a dose-dependent manner, or natural organic matter, alginine, cysteine, tannine, et cetera, were able to mitigate that toxicity. And we could see that heavy metal resistance genes, universal stress protein, antioxidant enzymes were able to mitigate the inhibition from trace metals. So when we talk about biodegradation, it's important to understand the mechanism at the bench scale, the pilot scale, and various in situ and ex situ applications, for all kinds of applications. We did develop tools to quantify fluorescence in the populations to assess if there was significant microbial isolates or microbial community that was responsible for dioxane biodegradation. We also focused on enzyme assays because it's monooxygenase, see if we could find surrogate assays. I showed one, in some of my early slides in which naphthalene oxidation was in some ways analogous to dioxane degradation in a limited number of monooxygenase isozymes. Some of my work also in about 6 or 7 years focused on compound-specific isotope analysis. We've published on that. Mike Hyman has a more recent paper. So CSIA is being developed as a monitoring tool to establish natural attenuation or enhanced biodegradation. And then in my lab, working with pure cultures, microcosms, as well as field samples, we do all kinds of genomics, transcriptomics, metabolomics, and things like that. So with CB1190, we submitted this organism to the Department of Energy's Joint Genome Institute, and they sequenced the genome for us. That was really nice. We were able to mine different type of genes, select which things would serve as putative biomarkers for monooxygenases, design those primers, amplified nucleic acids, as I said earlier, in pure cultures, microcosms, and in situ field experiments. So when we mine the genome, it's a really large genome for a prokaryote, almost 7.5 megabase pairs. And we found that there were over 2,200 sequences, open reading frames, that could be putative monooxygenases. So we down selected them. We first got about 20 or so, and then within that, as we were doing amplifying selectively in this organism, as well creating composite consensus primers for some of the other more naturally occurring sequences in the environment. So we focused on a few targets that are been shown to metabolize or co-metabolize dioxane. And first we thought we could find that cluster if the certain kind of taxa are present in an environment, then those could be closely related for dioxane biodegradation, as has been shown for chlorinated solvents, dihalococoides, some other very selected environmental biodegradation processes. Turns out in this case, dioxane degrading activity was kind of spread all over the prokaryotic domain. So we gave up on that rather quickly and chose to focus on the functional genes, or the genes coding for enzymes that carry out those reactions. And turns out that that tree was a lot more compact. And we found this clade over here indicated in purple, where most of the dioxane metabolizing or co-metabolizing strains and their sequences, the functional monooxygenate sequences, were very conserved here. So that was helpful to generate the primer sequences. We had some divergence with co-metabolism, but overall it seemed like a pretty tight sequence there. So we used that. We did several bench scale studies and studied the increase in gene abundance and gene expression, so transcripts, as dioxane was biodegraded. This is an example of pure culture. Nothing surprising here, 3 orders of magnitude increase of the functional genes as dioxane was degraded over about a week. It's important to note that the DNA and the RNA, so the genes and the transcripts, are good predictors of the protein formation, and then you can relate it to the degradation. Every once in a while, there are enzyme inhibitors, and some of the examples I talked about with the chlorinated solvents and metals that are post-transcriptional inhibitions. So they would not necessarily be captured by increase in the genes and the transcripts of dioxane degrading monooxygenases coding genes. So just need to be a little bit careful about potential inhibition and how we interpret some of these biomarker data. Here are examples from a paper in Applied Environmental Microbiology in 2014. When dioxane was degraded, some of the transcripts were actually downregulated. And that made sense because we were looking at a certain type of gene transcript. That gene is induced by dioxane itself. So when dioxane is rapidly degraded, the inducing pressure is gone. In the presence of an inhibitor, in this case, acetylene gas that completely is a suicide substrate for monooxygenase enzymes, we saw biomarker numbers were higher, but dioxane degradation was experienced a lag. So in real samples, there's a little bit of nuanced interpretation, but we're actually pretty happy that the biomarker sequences are available to us. I developed them, and then I just shared the sequences with a company, Microbial Insights, that is providing this service commercially. We also use that in one of the Air Force funded projects. In fact, Hunter is a co-author on this paper. Again, in a monitoring well, we saw dioxane degradation. In some of the microcosms, we added more dioxane. So when dioxane is present, we see this blue curve, which codes for dioxane monooxygenase. So DXMO enzyme, or the gene coding for that, DXM subunit B, responds to the presence of dioxane. So very specific biomarker. And then we amplify the gene that codes for aldehyde dehydrogenase. which is induced by the products of dioxane degradation. So that's a little more confirmatory biomarker taken together with DXMB. So DXMB and ALDH taken together would provide good, strong evidence of dioxane biodegradation. These are composite data. It's a paper we are preparing to submit, in which we took different studies, different amounts of co-contaminant level, different amounts of dioxane, ranging from single-digit parts per billion to tens of parts per million, different slopes of degradation. And we have, as far as field data go, at least I know 0.64 R squared doesn't look that exciting, but it was something that in early development of these biomarkers, we were able to establish that, yes, we may be able to quantify mass of dioxane degraded, not necessarily instantaneous concentrations, but mass removed by looking at the fold change of these biomarker abundances and transcripts. And one last little data point, I think I have about 3 minutes left. This is a project that we did, also published in Remediation Journal in 2016. There was a site that was a landfill, and dioxane contamination was resulting from that source. Curiously, chlorinated solvents were not detected at this site, but dioxane was. And over 6 years, dioxane plume, so the source is in the bottom right, and the plume is migrating in the northwesterly direction, if you will. So the plume was shrinking, and there were no active activities, any remediation activities that were being conducted. So we were asked to quantify biomarkers and see if we could use that as evidence of dioxane biodegradation. And indeed, we collected soil and groundwater samples, extracted nucleic acids without doing any lab bench scale studies, just straight PCR, qPCR them. And here what you're seeing, where we have red circles here, are dioxane monooxygenase detections or genes coding for that. The yellow would be aldehyde dehydrogenase, green would be both. We also looked at some co-metabolism powered by methane, because it's down gradient of a landfill site. So those are also data separately. But in this case, the presence of these gene biomarkers really tracked very well with the receding plume. So that provides some substantial evidence of dioxane natural attenuation here. So to summarize, yes, now we have a set of genes that could serve as biomarkers for biodegradation. We do think that if we are able to detect these functional markers, we may be able to identify and quantify indigenous dioxane metabolizing microbes. It is influenced by co-contaminants, so the care must be taken when we interpret these data. They would be useful in making some site interventions or natural attenuation decisions. And I would leave with that I get asked this all the time: are there biomarkers for co-metabolism? And I would say yes, but they are sort of indirect, because co-metabolism is a biological accident. You really can't design for it, but statistically you can use those data of propane or isobutane or methane or ethene monooxygenases as one of the lines of evidence to validate dioxane biodegradation. Some of our work is very fundamental and mechanistic, but we also work with collaborators, practitioners, as well as other researchers to translate this work in the field. And we've had some really good projects to report from there. And I want to thank federal government sponsors, industrial sponsors, partners, collaborators for their support, and happy to answer questions later. I will be available through the end of the meeting today.